Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
All right, I think most people have now made their way in, so I'll just do a brief plug for next month's webinar. So next month we'll have Ellen Zong at Princeton. We'll be talking about Dragon AI. So if you're interested in cool things in cryo-EM for dealing with heterogeneity, come back for that. And again, one final time, for any questions you have, just put them in the chat or the Q&A and we'll get them to Alistair at the end. And now I guess it's a good time to introduce Alistair Burt, who's now at Genentech, and before that was at LMB, doing a lot of cool tomography stuff at the time in Reliant 5, and now working on the warp pipeline for cryo-ET, which we've had a lot of questions about. We have a lot of users using, and it seems to be extremely powerful. So I'm kind of interested to see what's going on with it and where it's going to go in the future. So with that, take it away, Alistair.
All right, thank you very much, Sean. And again, I already said thank you to everyone for coming, but yeah, I really do appreciate it. My name's Alistair. I work in Dmitry Teganov's group at Genentech. So no longer in the academic setting, in an industrial setting, but still thankfully get to make the work that we do open and available to everyone. So I'll give a quick one-slide intro to what cryo-EM looks like at Genentech. So when I first started, I guess I naively assumed that everything in industry would be done at industrial scale. And then I've been super impressed to see how effective you can run a cryo-EM facility within an industrial setting. So cryo-EM was established by Claudio Sferi back in 2015 at Genentech, and it's now run by Alexei Rohu. We've got three people who run more on the kind of system operations and system specialist roles. So that's Matt, Kaylee, and Przemek. Przemek is the one who's kind of spearheading tomography efforts within the company. And then on the computational side, we've got Dimitri and I, and we develop Warp, which is the thing that I'll be talking to you about today. So, oh, my arrows are not working, so I have to click to go between slides. Just super quick introduction for anyone in case you're not super familiar with cryo-ET. So cryo-ET is this experiment you can do on a transmission electron microscope where you have a sample on a standard electron microscopy grid. You rotate it usually around a single axis, plus minus 60 degrees in 3-degree increments is the kind of standard experimental scheme. And then you collect these projection images at the bottom, and then you're able to do 3D reconstructions of more complex biological objects than you might be able to image and reconstruct using something like single particle analysis. In terms of doing structural biology with cryo-ET, which is kind of what the Warp pipeline is focused around doing, getting high resolution reconstructions from your cryo-ET data, I like to split it down into these three little blocks. So we've got a whole pre-processing block, which is everything that takes us from the raw multi-frame micrograph data that comes off the microscope to our nice 3D pictures that we like to kind of ogle over, these tomograms. Then we have this block that we'll call particle picking, where we have to find initial candidate poses for each particle that we might be interested in understanding a little bit better in our data. And then we also usually need some kind of initial reference that we'll use to bootstrap our way towards a nice high resolution reconstruction. And then once we've got those initial poses and a reference structure, there are various packages and things that you can do to optimize both those poses and the reconstruction that you get out. So we'll talk a little bit more in detail about those things as we move forward. So a first good question or a good kind of starting point to think about things is what has historically limited resolution in subtomograph averaging? So I really should get around to updating this plot, and I've said this on multiple occasions when I've presented this plot now, but you can really see that despite more and more people doing tomography over time, the amount of high resolution structures that are coming out has stayed relatively low. I think this has improved now, and we would see better numbers if we were to look at the modern data, but this trend is still there. Tomography is still hard to do, and you won't get high resolution from all of your targets. So the two things that I think are the main historical limiting factors are accurate treatment of the CTF. And so when we have images forming in the electron microscope as the result of interference of the scattered and unscattered electron beams, the beams that scatter to different angles will have slightly different path lengths. So it's just traveling a longer distance. And then as a result, when they interfere, you get at some points constructive and at some points destructive interference. And as a function of defocus at the distance of the sample from the focal plane, the character of this contrast transfer function changes quite a lot. And so here we've got between 1 and 2 microns at 2 angstroms per pixel, some simulated contrast transfer functions, and you can see that they oscillate a lot faster as you move to higher defocus. The sort of canonical first implementation that really showed that this was a usable approach for dealing with the CTF in tomograms properly and carefully was presented by Beata Turanova back in 2017. And they showed this nice approach for applying the defocus dependent on the position of the particle in the tomogram when they're reconstructing the tomogram. And this kind of general family of approaches is referred to as 3D CTF correction. And importantly, they demonstrated that this was able to increase the resolution of reconstructions of these HIV capsid hexamers down to around 3.4 angstroms, which is where you start to see side chains. And so it really was key in showing the promise of tomography as a technique that could move to these resolutions. But as I showed before, this wasn't the thing that really pushed us all the way there and got us more routinely doing these higher resolution reconstructions. So the other main limiting factor has been errors in projection geometry or reconstruction geometry. And so I'm just going to show quickly. I spent last night making these animations. So we've got a little simulated ribosome here at 4 angstroms per pixel. You can see we've got lots of nice fine details. You can see the RNA helices and things. And then, you know, if we can make a perfect tilt series of this ribosome and then a perfect tomogram of that ribosome, you can see that actually the reconstruction quality is really, really, really good, even though you have this missing wedge, this region of Fourier space where you have no information. The fine details are still there and the ribosome is pretty well reconstructed. And so you can imagine if you could average lots of those, you would get a very good high resolution reconstruction of your object of interest from that data. The problem is that when you start to introduce errors into the just the shifts in this case in your tilt series, you can see that as these errors increase in magnitude, you can imagine that the reconstruction quality would drop off quite a lot. And so here we've gone up to just a 1 nanometer average error in the shifts, and you can already see that on the scale of the tiny objects that we're imaging, this error becomes quite important. Here I've done the 3D reconstructions from particles with those different errors in the shifts, or the tilt series with those different errors in the shifts. And so here you can see, as we get towards that kind of just 1 nanometer of average error in the shifts, the reconstruction quality really drops off. You have none of that fine structure that we wanted and appreciated. And so again, just to reiterate, when you average particles that do not look, are not well aligned, you will be severely limited in the resolutions that you'll be able to obtain. And so the software package that I work on now is this Warp and M workflow from Dmitry Teganov, who's my boss here at Genentech. So he's been developing this for a really long time, about 10 years. And yeah, I'll take you through kind of the major steps of this thing. So at the very beginning, we have data that's acquired on the microscope. So we have these raw frame series or tilt movies. We then move into this kind of pre-processing block that I introduced before. So everything that takes us from these multi-frame micrographs to tomograms. Warp has nice, fast, very correct GPU implementations of motion correction, CTF estimation. There's what was one of the first actually neural network-based particle picking methods, or CNN-based particle picking methods, which is called BoxNet. We interface with the standard programs for doing tilt series alignment, so that's iMod and RTOMO now. We provide routines for tomogram reconstruction and also denoising of tomograms, so there's noise-to-noise based denoising. Then for particle picking, we only actually provide a template matching routine within Warp. Now, template matching is good for ribosomes or apoferritins or big, large, easily identifiable objects in your tomograms, but there's this old tweet from Dimitri. Even he is clearly not happy with how template matching performs when you're actually trying to solve a bunch of the problems finding particles in amongst all the complex biology that we've got going on in these tomograms. And so here's an example of this is actually something that I did during my PhD, which was kind of creating a nice interface between Dynamo, which is a package that provides a lot of nice tools for picking or annotating geometrical features within your tomograms, and then seeding particle picks with positions and orientations based on those geometries that you've annotated. And this is what allowed me to solve the problems of the data that I had during my PhD. And so it's an approach that I still recommend for people who are struggling and maybe not getting the most out of these automated approaches. If you have geometry in your system that you can leverage during your averaging, then you really should not be throwing away that useful prior information. It can really help guide your refinements. And so we then move on to this refinement block. And so Warp doesn't provide any kind of engine for doing these global searches over orientations. Instead, we export particles which are compatible with Relion. Over there, if you're doing frame series data, you can do 2D classification, and if you're doing 3D, you can do 3D classification, 3D refinement, ab initio, all of the nice Relion jobs that we are used to. Obviously, all those other jobs are also working for 2D data. Once you've got kind of initial particle poses and references, in tomography, you're likely to still be a bit limited by these errors in the shifts that we were talking about before, and there are also potentially still errors in the CTF parameters. And so, this is just a slide to illustrate the way these iterative refinement procedures generally work. You have sub-volumes from your tomograms, which are, the particular particle can be oriented in any direction in any particular instance of that particle in the tomogram. And then you iteratively align them to a common reference, average all of those particles, and then update the reference, and then iterate this procedure until hopefully you've got a nice shiny reconstruction. Once you've got this though, because of the limitation of the initial quality of your tilt series alignments and any errors in the CTF parameters, we need something that will kind of wiggle everything into place. And so the solution in our workflow for this is this program called M. So M is responsible for doing simultaneous refinement of both deformation models. So the images, we allow them to kind of warp and shift into place. So that's each image contributing to each particle will move a little bit just to kind of make sure that everything matches the reference as best as it can. And then, also the CTF parameters will be updated. And this is all based on understanding where your particles are within your multi-particle system. So M has this ability to co-refine multiple different, what they call species within your tomograms. And so you can have as many different types of biological objects as you want, as long as you know the position, orientation, and what that 3D structure looks like. Then M will use that information to optimize its description of all of the parameters that go into the reconstructions. And hopefully this significantly improves the quality of the reconstruction that you get out at the end. And so the specific parameters that you have access to when you're doing refinements in M are basic stage movements. So these are the simple 2D shifts of the images. You just move them over, and globally everything should match a little bit better. You can refine the stage angles a little bit. So do we really trust that 60 degrees is exactly perfect on the microscope? I think it's pretty good and pretty close to perfect, but you have the option of also refining those stage angles a little bit, and that might make a small difference. This two-dimensional image warping is something that we see benefiting quite a lot. And so if you we know from single particle cryo-EM that as you expose these thin electron transparent frozen samples to an electron beam, this exposure induces motion in the sample. And so by modeling a 2D deformation of the images, we allow for correcting for some of that motion in the final averages. And so that's really useful. You can't capture all deformations that happen in the 3D space of your sample as 2D image warping deformations. And so there's also this volume space warping. And then specifically for working with normal frame series data, so this is 2D data, not tomography data, also allows you to model this doming that people sometimes talk about. So specifically the movement is, your ice sheet domes a little bit as a function of exposure. M optimizes the parameters of this system together, and so you get a better model of where everything is, which orientation it's pointing in, and how that all contributes to best matching the experimental data that you have. So the first significant result from M, and the reason a lot of people got excited about trying it out when it was first released, was because Dimitri worked with Yulia Mohammed's lab to work on these Mycoplasma pneumoniae cells, and they had some of them that had been treated with chloramphenicol, and they were able to push the resolution of this reconstruction all the way down to the Nyquist limit of the data, basically, which was about 3.4, 3.5 angstroms in this case. And here you can see the drug molecule, chloramphenicol, resolved at 3.5 angstroms directly within the active site of this ribosome. And this is kind of the first demonstration of the real promise of in situ structural biology at higher resolutions. And so people rightfully got quite excited about all the possibilities and the things this might make possible. So hopefully that's given you a kind of good sense of the overall workflow that we have in place. The problem is that historically, Warp and M have been locked inside a Windows box. So Warp was a piece of software that was originally developed to run alongside the microscope. And for anyone who's used it, you'll probably remember it has this really nice graphical interface, and you can see, how defocus changes throughout your acquisition, how the numbers of particles are changing in different areas of the grid, and all of these things. It was a very reasonable decision to develop Warp for Windows because the microscope PCs also run on Windows. Having Warp and M kind of stay there when the rest of everyone's compute infrastructure, for the most part, was in Linux environments, meant that lots of people were quite frustrated about having to buy Windows machines to be able to test out and run this thing. And I think it slowed adoption quite significantly. And so the work that we've been doing, Dimitri, since before I arrived at Genentech, and then joined by me more recently, has been to unlock Warp from this Windows cage. And so Warp, the latest version, is now available on Linux. For people who are familiar with and don't have SPGrid, we provide a Conda package, which should make this installation relatively painless. There still might be small issues. If you come across issues, please do reach out. But we have tried to make this relatively easy, and we do appreciate that it's not always setting up lots of pieces of software is a frustrating part of doing this work for lots of people. So something that I know I really appreciated about Warp when it was first released was that there was good documentation for at least the core workflow. And this really did allow you to get started and explore and understand the way that things were supposed to work in the best cases. And so we've tried to reproduce that with the Linux version. If you go online, the website is warpem.github.io/warp. You can find under the user guide section, we've got this tutorial for processing tilt series data that will take you all the way from the raw data to a 3 angstrom map of apoferritin. Now, I really do recommend that people spend a little bit of time with the tutorial. It is obviously not that exciting to get, just another apopheritin structure, but it really does show you how the process is supposed to go when your data are really good, and that can be really informative if your own data are more challenging and not working for whatever reason. So you'll be much more likely to pick up on small user errors throughout that process if you're aware of how things should go in the best cases, and then you can kind of figure out just the parts that are specifically causing you trouble rather than just having a big amorphous blob of things not working and you not really understanding the workflow well enough to be able to troubleshoot these things. So yeah, any feedback on that documentation, please do drop an issue in the GitHub issues or on the mailing list. We also have been running workshops for users who don't like to read documentation. Basically, when we run these workshops, we do just run through the tutorial, and obviously it's an animated experience, and it's kind of a specific time for people to spend time on learning the thing, which is valuable. I know we're all super busy. So yeah, keep an eye out for announcements of workshops in various places, and hopefully you'll get to attend if you think that would be useful for you. But if you don't, the tutorial is there, and we do respond to questions online. So, you're not excluded. Brilliant. I'm now going to switch gears a little bit. So the Warp command line version of Linux was released about a year ago now, and Dimitri has been plugging away, or we've been plugging away on this thing that's called Relay, which is the web interface for these command line tools. There's obviously lots of good reasons for wanting these tools to be accessible through a nice web interface. One experience that I definitely had when I started using Warp on the command line was that the lack of visual feedback is not always ideal. So, tomography is this very visual discipline. We have these nice 3D images. We want to understand where our particles are in space and how they relate to each other and all these things. And when you are locked behind a command line and, maybe your data's away on some remote high performance computing environment or something, it makes it harder than it should be to go in and explore those things sometimes. And so, the web is the way that makes these things the most accessible and easiest to use from anyone's computer. And so we've been working on this web app, Relay. Dimitri actually, over the Christmas break this year, this is a picture of him in a hammock, and he rewrote the thing basically from scratch because there were various issues with the first iteration. And what you'll be seeing now are kind of screenshots from the current working version that we have. So it's a web application. It has a whole kind of user authentication system like you might expect. Here we're going to log in as an admin. Boop, boop, boop. And then once you drop in, you'll see that we have a kind of project management set of tools also within the application. And so your work is divided into projects. Projects are supposed to be these very long-running things. So it might be a whole kind of research arc within a lab. Maybe you're interested in a particular protein or set of proteins that form a specific biological system, and that might span the work of multiple PhD students or postdocs over time. Within there, we have these things called spaces, and so these are kind of specific locations on disk where you might store, say, a bunch of related processing for data for a set of data collections, maybe done by one student or one postdoc over a longer period of time. And then within spaces, we have these things called views. And so you can have any sort of logical set of jobs grouped together. Maybe it's all of your processing down one particular branch because you were interested in one specific, trying to do a certain type of classification, or maybe it's one view per one data collection. You can organize these things kind of however you want, but we have these multiple levels of organization. So within the view here, we've imported a frame series data set, and then we're running motion and CTF correction here. So you can see the kind of flow for building the job and then launching it. Once you've launched it, you'll see a card for that job, and you'll be able to see kind of how far things have progressed through all the items that needed to be processed, and then an estimate of the remaining runtime. Over here, we've got the visualization of the compute queues. And so over here, you can see that the job is running in the general queue, and you'd be able to see if you had a bunch of jobs stacked up and maybe things aren't flowing on the cluster or whatever is happening there. It's a nice way of kind of visualizing that. And then in terms of actually how you interface with, say, your local HPC environment or whatever computing environment you have locally, maybe it's just a workstation. There's a kind of inbuilt interactive editor for job submission scripts and these kind of things that let you get the environment set up and make sure that it works in your system. And so that part is delegated to the people who are managing your installation of Relay. Here you can see kind of what happens when you look inside this Motion and CTF job that we launched before. And so you can see kind of, for people who are familiar with Desktop Warp, it should feel quite similar. You can go and see all the parameters that you might want to see for that particular data set, and visualize the images, deselect things if you need to. Here, when you click on any individual image within that thumbnail panel at the top, you'll see these nice overview statistics that you might be used to. Visualize the image itself with various filters and things, and then also the quality of the CTF fits as a function of spatial frequency. Here now, we're looking at tilt series data set. So once we've imported these MDoc metadata files that tell you how all of the individual images within a tilt series are connected, you have this tilt series overview, and you can see, how many tilts we have per tilt series is the only thing we have in this overview panel here. And if you hover on an individual tilt series, you can see all of these little thumbnails for every image. And then if you click on any individual tilt series, you'll be able to scan through each of these images in a bit more detail and see a bit more detail about the per image parameters or defocus fits, motion per image. All these kind of statistics are visible in this panel. Here's what things look like when you run these tilt series alignments. So this is an example of a job where we delegate out to an external package, in this case, eTomo from iMod, which does either patch tracking or fiducial-based tilt series alignments. And so, you can see some statistics from those alignments. And if you wanted to, you'd be able to visualize the result of applying the transforms to the 2D images, which align them and then hopefully allow you to make a nice, good reconstruction. And so you'd be able to visualize here if an alignment had sort of worked, or if it seems like you might need to play with the parameters a little bit to get something that's performing well for your data. We also have a way of visualizing tomograms in the system. And so here you can see the nice little apoferritins from the tutorial that we've been running. You have X, Y, and Z ortho slices all rendered next to each other here. And you can see, in this case, we see that the sample has been a bit tilted in the microscope, but the actual quality of the reconstruction itself looks really good. And then here's an example of a refinement job running. And so, as I explained before, in WarpNM, we kind of delegate the refinement block to Relion, and we've built this nice way of visualizing the results. And so you can see as the refinement progresses, how the map changes, how the distribution of particles changes, both in terms of number of particles in each class, but then also how the distribution of orientations within each class changes. So you can kind of use this information to identify problems and then hopefully guide your decision making as you progress with your own data processing. And that's about everything that we wanted to show you guys for today. I'll finish on one final note, which is that tomography is still really difficult. Having the existing set of tools that we have nicely workflowified will definitely make things easier for everyone trying to practice, and especially people who are newer and people who are maybe a little bit less computationally savvy should hopefully have a much easier time. That's what we've been trying to do here. But we're still looking at difficult problems. The the samples that we have in our images are not just purified protein on a grid. We have all of this extra stuff going on that makes picking a lot harder. And you really do still need to be thinking critically about how we solve every individual problem that's presented by our own data. And I don't want to give the impression that, now that we've come along, everything will be easy and immediately solved for you. But hopefully we've provided a useful set of tools that will, simplify the parts that should be easy in the future. And then you can go and solve just the specific problems, and everything that should be easy is. So yeah, with that, thank you very much, everyone, for listening. If you've got any questions, I'd love to take them. And yeah, thank you for listening. I think you might be muted, Sean.
I am muted. Yep, so we've already got a couple of user questions in already, so I guess I'll just launch straight into those. So we have a question from Alex Noble saying classification is often the slowest part of a tomography project. So how fast and robust is classification in Relay?
So classification in Relay is classification in Relion. The major thing that's changed in recent history with how that works is Relion Shaw's implemented this nice ability to run refinements on 2D particle series data rather than these 3D subvolumes. And so that does make these jobs run a lot more quickly. I agree with you that I have not had great experiences trying to use classification with tomography data in various packages. It still feels like a not completely solved problem, or at least in my experience back when I was more actively trying to solve my own problems. The particle series thing definitely makes a big difference in terms of how robust it is. I think there's definitely still some parameter tuning that you need to do. I've heard lots of rumblings of people not feeling like things separate in the way that they should. When I do this work myself, I definitely don't treat it like single particle where I rely on classification to sort out my particle set. I try and work with the cleanest particle set possible from the beginning to preempt and solve those problems. Hope that answers the question.
Yeah, we have another one. This time again talking about the Relion integration. So when you're submitting a Relion job through the Relay interface, can you submit that just through the Relay interface, or do you still have to export the data, go through the Relion GUI, then link it back and kind of move back and forwards?
Yeah, so that integration is very tight, even with the interface. And so you submit those jobs and look at the results and everything through the interface. There's no clunky run scripts to convert things or anything like this. Relion is the primary refinement engine that we use, and we want refinement to be a part of the workflow that you get when you run our software.
I guess related to that, how are you interfacing particle picking into Relay? Because I'm assuming it's still going to be using the warp template matching for tomography, but a lot of the time people are picking their particles in other packages. Do you have a good mechanism for loading that data in and visualizing it within Relay?
The unfortunate reality is that the lack of standardization of the way these data look and the way that everyone's data looks on disk means that we can't do a great job of being able to import any particles from anywhere. And so the approach that we take is that we say, we accept data that looks like this, and then I can help write a conversion script or whatever it is from whichever software package people are using, if it's Crayola or if it's Dynamo or whatever that thing is. But we don't explicitly have nice import routines for this thing. It's definitely a piece of the puzzle that's still left up to the user for now. I would love to spend time developing nice 3D annotation tools in the web interface, but that's a pretty big commitment and project, and it's not one that we've taken on, at least for now.
Okay. So does it include the kind of what is it called, Napavi Tomo slice style and warp template visualization tool that's kind of built into the interface as it is though?
Yeah, no. So again, those applications that you're talking about are desktop applications, which unfortunately means that integrating them into a web browser is not great. The parallel that you might be able to draw if you're used to single particle work is Chimera X is not well integrated into the CryoSpark user experience, but it is still the standard workflow for doing these things. And so, yeah, if you wanted to do your particle picking in any of those programs, you could, but you would have to bring your data locally first and then do your picking there. Thank you for the Napari Thomas Slice plug as well. Yeah, this is if you haven't heard about this for people, it's a Napari-based tomogram picking package that I've been writing with a PhD student from Göttingen called Aisha, Aisha UNL, and she has done a great job of integrating these picking workflows for picking filaments and spheres and surfaces, and that is a solution that you could go to, works similarly to Dynamo, but it's not in a MATLAB box, and so might be preferable for you.
So we have lots of questions coming in, so let's catch up on some of these. So is the CTF refinement and V-alignment in Relion also included in the VLay pipeline?
No. So the way that we use Relion specifically as a refinement engine, and when I say refinement engine, I mean it does these global refinements, so looking for orientations. The particles themselves are just moving around, but you're not doing any of the move the individual images that contribute to each particle kind of refinements. We have an engine for that ourselves in M. It works really, really, really well, and we don't, yeah, we like using Relion as a refinement engine for the other pieces, but we like M more for all of the nice high resolution polishing step at the end.
Yeah, so you're essentially doing the same fundamental conceptual workflow. You're just doing it within M rather than within Relion.
Yeah, M treats all of those final refinements in one optimization procedure, which is M. Relion treats it as a couple of separate programs where you've got one that will try and do the motion and one that will try and do the CTF refinement. Something that I think maybe this is changing, maybe this has changed. I haven't kept up super well on how things have been changing in Relion, but I don't remember there being a good way to do reconstructions of tomograms that have been improved by these refinement procedures. So Relion, my experience was that it was quite focused on trying to give you this nice high resolution reconstruction of your particle, but less concerned about improving the tomogram itself so that you could then, have a better time picking rare species or whatever you might be working on. And so there are some subtle differences, but you're right that conceptually it's broadly trying to do the same sorts of refinements. It's reference-based correction of motion and CTF.
So we have a question again on, again, kind of web UI viewing. So they say the iMod slicer window is great for viewing practically any angle of a tomogram, which isn't available in really any other package. How feasible is it to implement that kind of angle slicing and viewing into VLay?
It is feasible. It's a project, it's a thing that would take some amount of time. There is no fundamental reason that you cannot do this. There are very high performance, nice web technologies for doing 2D and 3D visualization. The one that I like the most is this thing called Molstar for doing molecular visualization, and that has support for viewing arbitrarily oriented slices. Yeah, it's a thing I would also really like and appreciate the need for, but unfortunately it's just not there yet. But you'll be able to download maps and tomograms as you would expect and look at them in whatever package you would like to. You're not confined to the web GUI just because we provide a web GUI, and hopefully over time there will be more and more things for you to play with within that web GUI.
Nice. I guess based on that, given I'm mildly inherently suspicious of some GUIs, I'm assuming all of the functionality that is held within the VLay web GUI is still going to be accessible via command line through the existing Warp M tools set.
Yeah, so everything that is being run by the web GUI it's running a Warp Tools program or it's running a Relion program in those instances. So the way we run iMod and RTOMA is also through Warp Tools wrapper programs that then put the data in the way that we expect for the rest of the Warp Tools processing. That's not going to change. You will still be able to go in and, if you want to use the command line for any particular thing, you can and are welcome to. It might not, well, it would not then populate the GUI automatically, though. So that's a thing that you should be aware of. If you want to be able to visualize things in the GUI, I think you need to process things from the GUI.
Okay, so it won't pick, even if you follow the same sort of directory structure, it wouldn't automatically pick up jobs that were run within that.
No, yeah, kind of the control of those things is done at a higher level and is controlled by the web GUI rather than by Warp itself.
This is maybe slightly too technical a question for this, but does that mean the way of handling the settings files and the various XML settings is changed from the current Warp tool where all of the kind of job-to-job settings are carried over in those XML files, and it can be quite difficult to go back and fork off from earlier in the workflow?
Yeah, so the settings files are still a concept, but you are not exposed to them nearly as much when you're interfacing with things through the web GUI. And so certain things, the only one that I think is it makes it more difficult to fork things is the inclusion of the tomogram dimensions in the tilt series settings file. Can you think of any other examples of things where you felt limited in your ability to fork because
I've sometimes found it difficult to go back several steps if I've realized that, 3 steps ago in the warp like setup, I wanted to change a parameter during tilt refinement or during motion refinement. I could find it a little bit difficult to keep both of those things simultaneously and be able to pick which one of those paths I want to follow for any given job.
Yeah, okay. So specifically for M refinement, yeah, you're right. We keep a history of the different versions of the metadata, but the way it works is as a Warp in general is a kind of state machine. It's constantly modifying this central set of metadata that describe the image or the tilt series as a multi-particle system. And then, yeah, the lack of a nice mechanism for reverting to older M refinements is definitely a thing that will be addressed in the web GUI. And so you will have a much more experience that you might be used to from other packages. You can fork off at any point and it will know the things it needs to and not presume things that it shouldn't. So you're not constantly modifying the same state in quite the same way. That's achieved through us wiring things up to make that invisible to you in the back.
Okay. Another question from the chat. What is the maximum resolution of the subtomogram average starting where you would recommend going into M for refinement?
That's a good
So how
question.
bad can it be to start?
How bad can it be to start? Yeah, I don't think it's extremely well characterized on the lower end how far you can go. So there will definitely be a point where the local refinements inside M will not be able to pull you in. And so I do think it's really important to try and start out with the best tilt series alignments you possibly can, and also the best set of particle positions and orientations. And you don't want to have a messy particle set where half of your particles are false positives when you take them into M. That will definitely make it perform less well. But you can also start at relatively low resolution. So I think people have started, if you're working with large particles like ribosomes, from 20 angstroms-ish and still been able to push things down to, kind of the 5-ish range. If you can get closer to 15, 10, things will probably be a little bit easier. But in terms of, yeah, try it out. You're only going to get as far as you get. You have to experiment. It depends on the strength of your data.
Great. Okay. I think we've used nearly half the time we've had so far just on questions. So there's a lot of interest in people in using this. I think we've got through most of the ones that I've seen. If other people have questions related to M, just drop them in the chat now because I'm probably going to start wrapping this up. But yeah, thanks, Alistair. That's really interesting. I know a lot of people are going to be very interested in this web GUI. I can't tell you how many times I've had people ask me when the equivalent of Tomo Spark is going to exist.
Yeah, I have had the same question many, many times over the years, and I actually always push back a little bit. And I do think, and this is personal opinion, that the diversity of the things that people are trying to do with their tomography data means that any one workflow is unlikely to solve all of your problems specifically. And the Warp and M workflow is a good example. It's quite focused on this producing high resolution structures outcome, but we don't really have a good handle or a good set of tools for helping you do segmentations and helping you use those segmentations to guide the way you search for particles. These are all things that we could think about doing, but it's not been the focus of development so far. And then, yeah, if it's unlikely that one package will solve all of your problems, you're kind of forced into a world where you have to use multiple things and integrate them. And yeah, I appreciate people wanting things to be simpler. I also want things to be simpler, but also make sure that you're still thinking critically about the problems that your data present and make sure that you are not just blindly following what the package is telling you it thinks you should be doing, because maybe your biological question doesn't actually need a high resolution structure. Maybe it needs something completely different that we don't provide tools for.
Yeah, I couldn't agree more with that. I did notice you're starting to include some segmentation tools. I think the latest M release included TARDIS, so you are starting to explore some other workflows, I guess. But there's no plan on this becoming a Scipion-like all-encompassing package which will do absolutely everything.
Yeah, no, I don't think that's what Dimitri is going for. I think he wants the place where you can run the tools that we have now been developing for, he's been developing for a really long time, and I've been helping him with more recently. We want these to be in people's hands and much more usable than they are, but I don't think he has ambitions for expanding to all possible tools in the near future.
Great. Well, I'm excited for the release of Relay. Hopefully that will come sooner rather than later. So for everyone who's left in the chat, I'm just going to wrap it up here and just say thank you again to Alistair for the update on what's going on with Warp and M. And again, to let people know that to come back, they should come back next month to hear Ellen Zong talk about some heterogeneity tools in Dragon AI. And yeah, I'm just going to stop the recording now and thank everybody for coming along.